The overall goal of this procedure is to detect and visualize waterborne Cryptosporidium and Giardia species. This is accomplished by first concentrating the water sample using tangential flow hollow fiber ultrafiltration. The second step is to elute the organisms and trap debris from the filter. Following a centrifugation step, immunomagnetic separation allows for selective separation of the parasites from the concentrated debris. Magnetic beads are detached from the parasites by heat dissociation and removed using a magnet. Cysts and oocysts are then placed on a slide, and the sample is stained for visualization by immunofluorescence microscopy.
The main advantage of hollow fiber ultrafiltration over existing techniques is that the ultrafilter is significantly less expensive and captures multiple microorganisms in addition to Cryptosporidium and Giardia, like viruses and bacteria.
Generally, individuals new to immunomagnetic separation will be challenged by the amount and type of debris associated with different samples from varied climate conditions.
Start by assembling the ultrafiltration apparatus inside a biosafety cabinet. Once assembled, the apparatus functions as follows. The water sample enters into the system by passing through the black T connector and moves through the pump head, the pressure gauge, and into the hollow fiber ultrafilter. The filtered water passes out of the side of the filter into the waste container. The retained water containing the organisms are forced into the retentate bottle and recirculated through the system until the volume of water is reduced.